Welcome to Microlab Mastery. Today's topic, Catalase Test. For a little background, catalase enzyme facilitates breakdown of cellular toxins produced during oxidative phosphorylation. Catalase enzyme neutralizes the bactericidal effects of hydrogen peroxide by breaking hydrogen peroxide down into water and oxygen gas. The Catalase test is an enzyme-based differential test for identification of bacteria. Catalase positive are generally aerobic and facultative anaerobic bacteria, while catalase negative are generally obligate anaerobic bacteria. This test is essential for differentiating catalase positive Microcacaceae from catalase negative Streptocacaceae, as well as other genera. There are two types of catalase test we will describe in this video: the Catalase Slide Test and the Catalase Tube Test. In order to begin the Catalase Slide Test, you're going to need the following materials: bacteria cultures, microscope slides, transfer pipettes, 3% hydrogen peroxide solution, and materials for aseptic technique, for example, an inoculating loop and Bunsen burner. In order to begin the Catalase Tube Test, you're going to need the following materials: bacteria cultures, transfer pipettes, and 3% hydrogen peroxide solution. Let us begin by describing the Catalase Slide Test. For the first step, please label your slide using a Sharpie or permanent marker. Step 2, please mix your bacteria culture using one of two methods: either by flicking the tube of broth, as is demonstrated here, or by using a vortex, as is demonstrated here. For step 3, please follow aseptic technique to sterilize a wire loop, as is demonstrated here. After you sterilize the wire loop, please ensure that the loop is cool before you place into culture in order to avoid killing the bacteria. Next, aseptically transfer bacteria from culture onto the slide. And please ensure that you follow aseptic technique by flaming the mouth of the test tube. Once again, please follow aseptic technique by sterilizing the wire loop. Then, repeat steps 3 through 5 for your second bacteria culture. Next, aseptically place 1 or 2 drops of 3% hydrogen peroxide onto each of your bacteria samples on the slide and immediately observe. Please note that when you observe, you will be looking for bubble formation, which will be due to oxygen gas that's released when catalase breaks down hydrogen peroxide into water and oxygen gas. In this example, bacteria A on the left was catalase negative because there was no bubble formation, while bacteria B on the right was catalase positive, which you note through the bubble formation. Finally, please properly dispose of bacteria into the hazardous waste container. An alternative to the Catalase Slide Test is the Catalase Tube Test. For the Catalase Tube Test, please add 0.5 to 1 milliliter of 3% hydrogen peroxide to each test tube and gently shake to mix. Please note that for the tube test, it is not necessary to follow proper aseptic technique because we will be disposing of the tubes when we are finished. Next, examine the tubes for bubble formation. In this example, on the left is catalase negative because there was no bubble formation, while on the right, the test tube is catalase positive because you can see bubble formation. Finally, please properly dispose of the bacteria cultures into the hazardous waste. This video has been a collaborative effort by Dr. Brian Sage and Dr. Dheeraj Verma. Until next time, this has been Microlab Mastery.